Welcome, everyone. On behalf of Cambridge Healthtech Institute's Global Web Symposia Series and our sponsor, Cellecta Incorporated, I'd like to welcome you to Molecular Snapshot with Driver Map Targeted Expression Profiling Assay. My name is Elizabeth Lamb, and I'll be the host and technical director for today's event. At this time, I'd like to introduce our presenter for today. She is Sunitha Sastry, PhD, Senior Product Manager for Cellecta Incorporated. Dr. Sastry, the presenter ball is yours. Welcome.
Thank you, Elizabeth. Good morning, good afternoon, and good evening to all of you who have joined us from different parts of the world. We are excited to be here. I'm going to present this. I'm going to take about 35 minutes to talk about this new technology, and perhaps we can discuss the rest, probably 15 minutes or so with your questions, and we will make sure we answer each one of them. Having said that, I want to start with introducing Cellecta, our company, and we are based in, and just to give you a little bit of an idea of the structure of this talk, this is what we are going to cover today. We are Cellecta. We are a custom service provider for functional genomics. We were founded in 2006, and we're headquartered in Mountain View, California. So we live in the beautiful San Francisco Bay Area. And we've been a solution provider for target discovery as well as biomarker discovery. These are the two portfolios. And let me just walk you through very briefly the target discovery tools and services that we offer. And this is in the space of gene knockdown, knockout solutions. And we offer complete screening services from start to finish. All we need is the set of gene list, whether it's genome-wide or a custom gene set, and your cell line of interest, and we'll do this project for you. We also provide reagents as pooled libraries, isogenic cell lines, and custom constructs. So these are a few of our offerings for gene knockout, knockdown, especially the RNAi CRISPR field. So this has been what Cellecta is known for. We have had extensive collaborations and publications. We're tracking quite a few of our reagent stream. And we're also extremely grateful to SBIR for all the grants. This has been, in 2015, we had the 12th SBIR grant. So this has definitely been our achievements for the past 9 years or so. Looking at what our team and our core expertise, we are a bunch of molecular biologists, geneticists, and genomic specialists. So we've looked at leveraging our expertise and moving into the genomics and transcriptomics services, and out of which I will be talking about Driver Map, which is our proprietary solution. And on this, just giving you a little bit of an idea of the red marks are just to give you an idea of what our proprietary technology is. With that, let me just walk you through today's presentation. And just so you know, most of these applications that I spoke about have been very oncology specific. So I'm going to use tumor microenvironment as an example, but certainly these platforms can be used for a variety of therapeutic indications. So as a sort of a context, providing you context to this talk, I just want to review a couple of challenges that we face that is driven by biology. So if you look at it, tumor heterogeneity is something that we all are aware of. Every patient, every tumor is unique. There's a complex interplay of tumor, immune cells, stromal cells, cytokines. So there are many, many players in this complex interplay. And when you look at the techniques that's available, there are still things that can be improved. High sample requirement, poor sensitivity, limited throughput are features that can be improved upon, and that's what technology providers like us are looking for. So in short, for people like Cellecta, what we are trying to do is provide you sophisticated tools, keeping these complexity in mind. So having said that, let's just review the concept of driver map. Now, there are many omics platforms to help you address some of the challenges that I talked in the previous slide. So here, this approach is a transcriptome-based profiling approach. Transcriptomics has always been the go-to for understanding the molecular components and the constituents of any particular cell line, your models of choice. So this is something that we want to make it as a transcriptome-based profiling approach. There are two components that we wanted to focus on, not only the differential gene expression, just getting an idea of either your set of genes or a genome-wide approach, but also looking at detecting cellular composition through gene signatures. So this is just giving you a concept, and I'll walk you through some of the validation and how we've looked at it. So from a technology perspective, in this transcriptome-based approaches, RT-PCR is extremely well known. It's been used. There are many techniques that have been prevalent for the past 30 years. So we have leveraged and adapted it to be our own, and it's an upstream multiplex RT-PCR and a downstream NGS readout. In our assay, what we've tried to do is get you information of about 8,000 genes in just one single reaction. So this is what we've tried to look at. This has been we are really proud to just look at such high multiplex feature. We've also looked at optimizing and making this platform broadly compatible, and also looked at reducing the amount of RNA that we can use in our assay. So having said that, for those of you who are very familiar with RT-PCR techniques, the biggest challenge or the biggest aspect of developing a platform is looking at primers. It's no easy task, and it's also something that has kept us very busy for the past 1.5 years. So if you look at that is the biggest component of how we looked at developing this assay, there is a design aspect to it. There's also a functional validation. So each of our gene-specific primer, we are looking at validating for specificity. So here's just a snapshot of few of the features that we look at for primers. So the first feature that we look for is specificity. So again, as I mentioned before, this is one gene out of the 8,000 genes. So you have to make sure that it is extremely specific to that gene of interest. So we start with 5 to 12 gene pairs, 5 to 12 primer pairs per gene, and then go on to optimize to what is the best performing based on specificity as well. We also look at sensitivity, and here's an example of some immune-related genes, and here's how we look at optimizing it with universal RNA from different sources. We also look at not just sensitivity. Sometimes if you look at just sensitivity as a feature, here are examples of genes which are extremely abundant. In technology such as this, you know that your reads can be completely eaten up by a few genes which are extremely abundant. So we also try to dial down the sensitivity and take some of these primers which have little medium performance rather than the highest performing primers. So this is where I am. Just to give you what I've tried to walk you through is very quickly give you an idea of primer design to prevent dimerization and functional validation of every single primer that we've used, and reaching up to the 8,000 mark as of now. And we are looking at specificity, sensitivity as some of the features in this platform. So on the second aspect, if you remember, I talked about the immune cell decomposition, or looking at deconstructing it to a specific immune cell. So here's an example of all the immune cell markers. If you look at, there are multiple markers for each of the immune cell. If you look at immunohistochemistry-based approaches, they are often very challenging and require a lot of tissues, but it also gives you a spatial information. So what we tried to do was give you a transcriptome-based approach that will give you a snapshot of the same. So if I could use just one slide, not getting into too much bioinformatics, I would use this. On the left is your hematopoietic immune cells. There are different immune cells. The higher the hierarchy is, it could be a much bigger gene signatures. So in this assay, we're looking at gene signatures. So a cytotoxic T cell, just to give you an example, could be a 60 gene signature to a multipotent stem cell could be almost a 500 gene signatures. On the right is expression profile. And then what we have spent a lot of time building is classifier models and algorithms. So what we've tried to do is put this data into our proprietary algorithm and try to see if we can separate a T cell from a mixture of all other cells. Can we look at a specific macrophage compared to all other cells? Can you look at NK cell and so on? So this is what has been both the design aspect, the bioinformatics aspect, as well as validation. So we could actually look to include as well as separate 38 modules of immune cells. Let me use another couple of slides to tell you why a high-sensitive platform is required for such an effort. So on the left, like I mentioned, cytotoxic T cell is a 60 gene signatures. But here, just to represent it, I've used about 9 genes as a representative data to plot it in a different way. So this experiment is an admixture, so where we've looked at adding FACS-sorted cytotoxic T cell into the mix of all other cells and tried to see if this driver map platform can pick it up. So you can pick it up at low levels. The signal to background becomes better as the concentration of this cytotoxic T cell is increasing. But it's also another feature that we try to optimize and make sure that our platform can not only detect is also reproducibility. So we looked at low RNA levels and tried to see if it's reproducible at low levels. So I know I've given you just a teaser of some of the validation, but this is what we have ended up in. Although we focused on the tumor microenvironment modules where we looked at cell lineage signatures, we also added immunity mechanisms, immunotherapy markers, all of these that are representative of giving you an information about different immune cells as well as mechanisms, and moved on to make this into a around 8,000 gene panel, which is definitely includes all the druggable genome, the clinically actionable genes, pathway information, and so on. So having said that, so what can it do? I'll just, in the next few slides, let me walk you through how this assay performs. So to start with, differential gene expression is one of the applications. You can use this platform for that. So we try to compare it with the gold standard. So RNA-Seq is definitely, as you all know, is the gold standard for this effort. So just to compare it, if your RNA-Seq is looking at 20,000 genes, this driver map is around 8,000. So we've tried to map all the genes that's overlapping between the two platforms. For the high abundance genes, there's a very high concordance, and driver map is able to show an increased sensitivity and also pick up a lot more genes because of its sensitivity. So the low performing genes can easily be picked up. So let me help you with this data. When you're looking at these two platforms head to head, for a highly abundant genes, that's not a problem. Irrespective of which platform it is, you could easily use it to obtain information. But the beauty of such platforms, targeted approaches, will definitely be in the low abundant genes. That's one slight difference that you would see. So the concordance will sort of change a little bit with the low abundant genes. But let's try and see how this, I've tried to compare it with an existing technology, which is a gold standard RNA-Seq. And here is an example to look at. On the left is IHC gold standard data, where you're looking at tumors being marked, you're looking at demarcating the immune cells. This is how this assay will show up, as in just looking at all of the genes and what would be the information, which is a quick snapshot meant to be at a higher level than the immunohistochemistry. So back to the story of immune cell components, this is how the assay would read. So on the left, if you look at a pathologist's report, you would probably have infiltrating lymphocytes or a few cell types being presented. And on this assay on the right, you can see all of these orange, as you can see dendritic cell lighting up, you have macrophages, T cells, and all of these cells. So I can show you this data. In the interest of time, I'm sort of running through this information. So now I've walked you through two information. One is differential gene expression, and the second is immune cell decomposition. And the third aspect of it, as I mentioned before, we've looked at validating each of these primers for specificity. So when we walk through each of these validation, they are not only tested for specificity, you can actually look at if they're validated for your cancer cell models. They're also looked at cross-reactivity, which is very, very important when you're looking at xenograft models. So this is just to give you an idea that this particular data is around 300 pan-cancer genes, and the data was generated using 24 cancer model, these regular cell lines that we use in oncology screening programs. So that's just one way of also using this particular platform. So I've come to the last few slides. What we've really looked at is making this platform broadly compatible. So we know we can use as little as 10 nanograms. It depends on the integrity of the RNA sample. Your input here is total RNA. So for blood-based biomarkers, for frozen, for any of these tissues, it can easily be dialed down to the 10 nanograms. For the FFP, again, integrity of the sample is our key, and it requires a little bit higher concentrations of total RNA on this. The other feature, the aspect that I'd like to talk about is when you're looking at blood-based markers, we don't need to do any ribosomal or globin depletion. So there are no modifications that can be directly used on this assay. So it sort of makes it very, very easy workflow for you all. So in a snapshot, if you look at platforms such as this that we have developed, driver map, we are using this name to talk about an end-to-end comprehensive service. So one of the applications that you can use this is for biomarker discovery. So if you're looking at, of course, this is a targeted panel and you're looking at differential gene expression, or let's say you're looking at a blood-based biomarkers, and you've identified a few genes. So what can Cellecta do as the next step? So here's where we also have additional capabilities of providing you a custom panel or taking it closer to your clinical assay. So where, for example, we could also develop a follow-up RT-PCR-based assay for you. So this is something that, because all of the primers that have been validated have been used, we could make that as a custom panel. This particular platform can also be used to look at molecular characterization of your cellular models or xenograft models. Or the aspect that we focused initially was on the tumor microenvironment. Because we're looking at immune components, it can also take us from therapeutic indication from cancer all the way to all the immune-related disorders. So that's another intended applications. So I'm here trying to summarize on my last slide so that we have enough time to talk about your questions. So this platform is meant to be high throughput. That's what we've tried to look at, profiling thousands of genes in one reaction. We're taking as a platform, we're moving towards genome-wide. And this is something that you're looking at one reaction, and hence throughput is extremely high. And that's also a cost-saving benefit. Sensitivity and specificity is what we've looked at. We've tried to look at low abundant genes as well as the very high abundant genes. So it gives you an idea to look at both. They're reproducible and reliable even at 10 nanograms of RNA, and it has broad sample compatibility. And as I mentioned before, these are some of the applications that we've focused on. With that, I'd like to thank my Cellecta team here, as well as some of the IHC studies, although I've used just one or two that was performed by our collaborator, Core Diagnostics, which is a CLIA lab, which we often use it for sample handling. And some contact information is provided, so if you have not been able to get your questions across, we'll be happy to walk you through and perhaps send an email answering your specific question. So with that, I'm just going to review some of the questions and try to see if we can get the answer. Yeah? So I have a question from one of the attendee saying, can driver map determine the abundance or the percentage of immune cell on a single slide? So it's a great question. The way we have developed this assay is to look at qualitative determination. So it is, for example, if you see a lot of IHC data, it'll say plus or plus plus. That's what is it is meant to do at this point. We are looking at, there are ways in which we can contribute to an enumeration-based assay, but it is something that we offer as a custom. So there's also another question talking about algorithms and if all of this is done by Cellecta. So when you're looking at this particular service, we offer it as a basic service and an extended service. So for a lot of bioinformatic analysis, we do have capabilities, but that is dependent on your project. And that is where we will try to look at it as a custom project so that we could include your bioinformatic analysis that you wanted. So, but we do have, to answer your question, we do have capabilities. Okay, so what kind of specimen is needed for a blood assay? So we, again, these are questions that we try to understand your workflow, and we try our best to work with you in what is available. So if you're looking at extraction of total RNA from blood, that is also something that we could use CLIA certified partners to handle specimens. But we also you can also send purified RNA either from whole blood or from PBMCs. I hope I've answered that question. Have you tried? Okay, so have you tried the same probe for cancerous and non-cancerous cells? I will check this information for you but we do have total RNA from different sources, from regular cell lines as well as cancer cell lines, but I'll have to check that information and get back to you. I do know who the question is coming from, so I can get that answer back to you. Is the assay automation compatible? Yes, we have an Illumina platform, and so we're using Illumina in our assay. But as a reagent, if you're interested in procuring an assay from us, we could also think of optimizing it for you. That's definitely the question. The answer is yes. Yeah. Can you comment on sensitivity? So in the context, so every platform, every assay provider would talk about sensitivity. So just to give you a context, if you're looking at infiltrating immune cells, they're usually around 0.5%, I believe, and we can certainly detect that. So that is the key to answering in the question in a different way. The other information is I can send you some very low abundant gene information. So sensitivity is certainly there in the platform that can pick up very low abundant genes as well. And that's what I meant as in when we compared it to RNA-Seq, we could actually get more number of genes in this assay. So I hope I've answered that question on sensitivity. Okay. So detection on minimal residual diseases. I am not very sure of one of the questions. The question was about detection of minimal residual disease. If I take it as if it can be looked at as a longitudinal time point, perhaps I'm reading the question wrong, but yes, if you're looking at levels of a biomarker changing across a period of time, that's something that we have built this assay to be sensitive to pick it up. But again, everything is dependent on that sample and that particular problem. Can you detect CD47 expression level? Yes, we can. Rare cancer cells in blood, that is something that is being optimized. That is definitely in making our blood-based assays, our assay to be more catered to blood-based biomarkers. So we're very happy to take collaborations at this point for blood-based work. So the answer is and the question was, can you detect rare cancer cells in blood? That is something that we would also like to be part of. So we could take this offline and perhaps discuss that project. I think I've come to the end of my questions. Again, I really want to thank each one of you for spending some time with Cellecta. We will be sending you information of this webinar, the recording, as well as the slides. We'll be happy to field any questions. We're excited with this platform, and we're really looking for your feedback. So please feel free to correspond with us and let us know what you think. Is this something that makes sense? Is it something that we should focus on? So please feel free to give us your feedback. Yeah. With that, Elizabeth, I really want to thank you for being there and walking me through this presentation. And to all of the participants, thanks for spending your morning or afternoon or evening with us.
Thank you very much, Dr. Sastry. And thank you, everyone, for attending. We're very grateful you came and spent some time with us today.